back, maybe a little too far. And Dr. Hashim is a postdoctoral fellow in his laboratory who's been working on this animal model. So thank you both.
I'll let Azat get set up. So I think as many of you know that we've had, so we know the gene that causes ALD, and we've had a mouse with a gene knockout for many years, going back to I think more than 20 years now. And the challenge has been to try and find an inflammatory model within that mouse that resembles cerebral ALD. And because that mouse does not develop brain inflammation like humans with ALD do. So we are, that's been one of the major challenges for working with the mouse model, is that we don't have a way to study the kind of inflammation that we see in ALD. So with that in mind, we had a conversation with Josh Bonkowski a few years ago, and he was working on an inflammatory model in the zebrafish. And we chatted a little bit, and we thought, well, we'll try this kind of a version of this that he was using in his zebrafish in our mouse model. And around the same time, Dr. Hashemi who was in a neighboring lab, had a space in her postdoc to come work on our lab, and she had been working on some inflammatory mouse models for MS, and she brought her knowledge to our lab, and she had some really key insights that made this work for our model. So I think I can say without Dr. Hashemi's initial insights, we wouldn't have the model that she's developed, and she's done a really wonderful job. We're excited about the potential of this model. It's still early stage, and I want to emphasize that it's a model. It's not human cerebral ALD, but it's a version of it. So I'm going to let Dr. Hashemi talk about what she's done.
Good morning, everyone, and thank you, Dr. Van Haren. So my talk is a mouse model of cerebral ALD. You know, a model for any disease is very important to understand the molecular mechanism, what's wrong with the molecular inside the cell or environmental of the cells. And when we understand this mechanism, it's very easy to find the target for therapy. That's why for cerebral ALD, if we have appropriate models, it really help us to find the treatment for the disease. You know, cerebral ALD is most of the finding coming from MRI. And here you can see the MRI with the lesions in the white matter and gray matter. And then mostly cerebral ALD is started at very young age. 40% at age 4-5 years, and 20% of ALD, that is a cerebral ALD, is in adult age. And almost 2/3 of the ALD patient develop cerebral ALD. And this cerebral ALD affect on the brain and adrenal. And there is a different phenotype of ALD. Why we have a different phenotype of ALD? There is just one mutation in ABCD1, but we have a different phenotype of ALD. And the phenotype is based on the different severity of the disease and different onset of the disease. Some start at very young age, some start at early, maybe 10 years, and some start at 20, 30 years of age. So why it happens? Sometimes mutation is just once mutation. Sometimes it's a big mutation inside the genome. Sometimes it's a big deletion of the part of the gene, but it cannot predict the phenotype of the disease. And the disease have different phenotype and makes it very complex to understanding the mechanism, and also have a good model for this disease. For example, this is a 5-year boy with the CLLD. That is very normal in MRI, and phenotype is completely normal. You see here, white matter is normal, But after 6 months, white matter, we can see lesion in white matter. This white spot is a lesion. And it's very progressive during the 6 months. But the phenotype is still normal, no disability. But after 12 months, disability is expressed and then phenotype expressed, and the lesion progressive very rapidly to the other part of the brain in white matter, gray matter, cortex, and this is a stage that disability, and then they cannot move and walk well. So these are the MRI findings. Then we wanted to see what is inside the brain, what is the histology inside the brain, what happened. This is based on this MRI. This is a human post-mortem tissue that we look at this area to see what happened in this area. Here, this part of the cortex close to the white matter, there is no myelin looks normal. There is a myelin here, but there is a lot of immune cells, mostly macroglia activation in this area. This is the area very active, the myelination happened here. You see, there is not blue. This is myelin loss, and a lot of immune cell infiltration we can see here. This is a vessel, that vessel like extended, and a lot of immune cells coming out of the vessel and go to the brain. That are the damaging cells. This is a B cell, T cell, monocyte coming out and make a damage. Here, this is our lab did, and we look at the one marker for ALD that we could see this immune cell, macrophage, monocyte, and sometimes astrocyte express IL-18. IL-18 is a factor of innate immunity. We have innate immunity and adaptive immunity. And in adaptive immunity, we have a B cell, T cell. That is, we can in peripheral blood. But here is innate immunity, the first defense of the body. That IL-18 is product of the innate immunity that we can see in macrophage, macroglia. And then this area is completely now myelin and completely demyelination, and there is no monocyte macrophage. Maybe because monocyte macrophage phagocyte myelin, and there is no myelin here, and all the macrophage monocyte, die because of the too much phagocytosis, or they don't migrate to this area. So these are the findings from MRI and histology. So based on this finding in human, we have early age onset of the disease. We have MRI with T2 and T1 gadolinium. Then we have activation of adaptive immune response, B cell, T cells. We have perivascular space. We have demyelination, we have geliosis, we have macrophage that have myelin inside the macrophage, IL-18 expression, and then axonal damage and neurodegeneration. These are the factors that we could see in human. Based on this factor, we want to develop our model. There is a different model for ALD, and like a cell model, fibroblast from a patient, there is a yeast, there is a C. elegans, Drosophila, and then zebrafish and mouse. They are very good model to help us to understand the mechanism, like accumulation of very long fatty acid or beta oxidation. But this is a disadvantage of this model because most of them, especially C. elegans and zebrafish, they don't have adaptive immune response. Even the mouse with ABCD1 mutation, they lack of inflammatory demyelination in the brain. It means mutation is necessary, but is not sufficient to make a lesion inside the brain. There is other signaling that activated this, with this mutation, activated inflammation in the brain. So what we did, we make a model of cerebral ALD. There are two models. One is cuprizone, one is experimental autoimmunity and encephalomyelopathy. These two models are multiple sclerosis model. They are not a CLLD model. They don't show any brain inflammation in the model. Cuprizone model, we give the mouse cuprizone in diet for 2 weeks. These are as a control. For these mice, we injected mice with part of protein, part of the myelin. MAG is a part of the myelin protein. And because cuprizone make a activation of innate immune response, EAE activated the adaptive immune response. Both these doesn't show any neuroinflammation in the brain, any lesion like cerebral ALD. So when we mix these two models, we give the mice cuprizone for 2 weeks, then injected MAG, that is a part of the myelin protein. And then after that, we collect the tissue. Here we activated innate and adaptive immune response in the body, in the brain. Then after that, we look at the clinical score, what is the behavior, and we look at histology. We do several staining to find adaptive immunity, innate immunity, demyelination, or also we do MRI to see if this model is similar to human cerebral ALD. So we don't know about this model. All of this for human, this is a question about this model. We go step by step to see if this model. Here is a EAE model. If you look at the EAE model, we have a wild type and we have an ABCD knockout mice. When we injected MAG or myelin, we couldn't see any difference between these two groups. And if we look at white matter like a human, this is the area that in human also is a similar involvement in demyelination. We couldn't see any macrophage, monocyte in this area. And there is no demyelination in this area. Then we do cuprizone induced cuprizone. And we could see some here, macrophage monocyte, but we could see a little bit demyelination when compared with the EAE. But when we do cuprizone EAE here, this is a behavior phenotype. We could see a little bit in 12 and 13 days significant difference between ABCD knockout mice and wild type mice. And when you compare, monocyte macrophage in this area, we have a lot of monocyte macrophage and we have a demyelination. See, the staining is weaker when compared these two in this area. Sorry. Yeah, this is an age. We have 2 age mice because ALD starts at young age. We wanted to see if age affected. These are 2 months, and these are 4 months mice. We could see at 2 months mice, we could see a lot of monocyte macrophage activation. Here you see in white matter, in thalamus, you can see the vessel, like a leaky vessel, and immune cell coming out from the vessel, and also macroglia activation. So it shows this model also age dependent. Yeah, this is a MRI T1 weighted with the gadolinium that we did, like a human. This is a human. You see, this is a lesion in this area. And this is a knockout mice in this model. We could see the lesion here like a human. And this is a naive, no cuprizone, no EAE induction. But you see white matter, we couldn't see any lesions. And this is a T2 MRI. In T2 MRI here, we could see the myelination. And here we could see, in knockout mice, we could see the myelination here. That is in normal without cuprizone or without MAG injection, we could see normal myelin. So This experiment shows, early stage of disease in these mice, and also higher demyelination and lesions in these mice compared to the control. This is like human almost. Then we look at the adaptive immune cells infiltration, B cell, T cells. The red is a T cell, the green is a B cells. It's coming out from corpus callosum. Here I show this is a mouse brain from this area. These are inside the ventricle and start penetrating to the parenchyma. This is a infiltration of immune cells. So we have also inflammatory infiltration of immune cell that is most we could see in knockout mouse more than wild type mice. Then we look at the this is a CD68 positive cell, marker of monocyte macrophage, and this is the DAPI, that's a marker shows all of the cells. And this, we merge these two together. We see this is a small cells around inside the vessel or around the vessel, and here the cells are around the vessel, start penetrating, but still they are around the vessel. We gave it grade 1, grade 2 based on the severity, and this is a grade 3. You see when the cells coming out from the vessel start migrated and penetrated to parenchymal more, and this is a very big lesions that we gave it grade 4. So I show it in this cartoon based on grade 1, you see, is a small lesion, bigger lesion, and then grade 4 is a large lesion in the brain, and this is a just normal, normal food we gave to the mice. This is, we just gave them cuprizone. Here we gave them just MAG injection. Here we gave them cuprizone and MAG injection, both. This is our model, this one. And we can see a lot of prevascular infiltration and with big lesion in this model. When we did a statistical analyzing, we saw this is a very significant. Then, based on that human, yes, we could see prevascular infiltration in this model. Then, galiosis. What is galiosis? Galiosis is macrophage and astrocyte inside the brain. Then, ALD patients, cerebral ALD patients shows galiosis. Then we look at exactly the same area that we could see lesion in the brain. This is a mouse brain. This is this area that we did a staining. And we could see very galiosis in our model. More astrocytes and more macrophage monocyte in this area. So based on that, we see reactive galiosis here in this model more than the normal. Then we look at the innate immunity. Okay, we know this adaptive immune response is active in this mice. We look at the innate immunity. This is IL-18 expression. CD68 is a marker for monocyte, and this is a product of innate immunity. Then we could see, these cells also express in cells in cerebral ALD mouse model. And this is a higher magnification. You see co-localization. This is a vessel, and we could see all the monocyte coming outside the vessel express IL-18. This means this monocyte, this macrophage are pro-inflammatory macrophage that coming out the vessel and going to the brain. This is a higher magnification that you can see co-localization around the vessel, even not around the vessel. In the parenchymal of brain, we could see IL-18 expression. This is also shows the vessel here, big vessel, IL-18, and then CD68, and this is the whole cells infiltration. Okay, so this mice is a very good model so far that they shows IL-18 expression like a human. Then here we could see prevascular space. In prevascular space, when the monocyte or macrophage from the blood coming to the brain, they start phagocyte myelin, they eat myelin. And we could see around the vessel there is no myelin. Even this is a big lesion. In big lesion, there is no myelin here. This green is all myelin staining. And these are monocyte macrophage staining. But the area that is full of macrophage monocyte, there is no myelin. And they phagocyte the myelin in the lesion. And then we did other staining for myelin to see if there is a demyelination, just confirm or a study for confirmation. And we could see here, like we must see in this model, like a grade 3 demyelination in the same area of corpus callosum that we could see in human. All right, so based on that, we could see confluent demyelination in this mice compared to the control. This is close to human. Then we look at the oligodendrocyte, because oligodendrocyte make a myelin. Then we count the oligodendrocyte, exactly the same area. We could see the loss of oligodendrocyte in this area. Yeah, this is like a human lesions. We see this criteria, and we could see this criteria in this model. And in human, in phagocytic cells, we could see myelin. We wanted to see, there is human lesions. This is a hippocampus, and these are, green is myelin. The red is phagocytic cells. And we could see a lot of phagocytic cells have a myelin inside. It means they're eating the myelin. Here is higher magnification. You see, this is a yellow, yellow spot here. There is a myelin inside this red cell that is a phagocytic cells. So these phagocytic cells has a, eating the myelin. So yeah, PLP positive macrophage also is like a human we could see in this mice. So this mice shows very good similarity with human lesions. And then there is a axonal damage and neurodegeneration. So we haven't done in this mice. We are doing this very soon to confirm all criteria this mice has is similar to the human lesions. So this model is very good because we can understand the molecular mechanism, why ABCD mutation happen, and then what's the serial events that cause the lesion in the brain. That is very good for understanding the mechanism, especially in neuroinflammation. And then also anti-inflammatory and help us to find in anti-inflammatory and neuroprotective treatment for progressive neurodegeneration. Is not only for cerebral AD, it helps for maybe other severe neuroinflammatory diseases. And we have a plan to target innate immunity in these mice to see if we can decrease the lesion, decrease the inflammatory, and decrease the demyelination in these patients. Yeah, this work couldn't be done without the help from this foundation, NIH, United Leukodystrophy Foundation, and other foundation from Max and Ben Memorial Foundation, and Dr. Van Haren helped a lot in this, Dr. Masilino, and all my lab members in Dr. Van Haren lab helped to do this experiment. Thank you so much for attention.
Thanks to the ULF for funding. Luann, did you have a question? Sure, go ahead. Yeah, well, the mice are available anywhere. The model will be available anywhere as well. So it won't be limited to one university, but we'll be working with it we're already working with it with a few therapies now. So looking forward to reporting more on that soon. Great. Well, thanks everyone.